got the sinuates. Use the ones that I've already given. Some of those of course they are correct the way you have given them. Hiyo lobe huwa tunaitumia at the apex. So always use it at the apex. Avoid using it within the margin. Someone was asking that I should emphasize on the type of leaves. I want to go there. Types of leaves. In fact, it has been illustrated within your screen there. The simple leaf. The simple leaf is normally single. Like now, if I take a leaf from the maize plant, that is a simple leaf. If I take a leaf from the bean plant, that is also a simple leaf. Then palmate, wenye walikuwa wanauliza ndio hiyo, you can be able to see it has got 5 leaflets within it. The trifoliate iko hapo, Hakim amewawekea. Pinnately lobed, palmately lobed, pinnate, it's okay. I think that is whatever I wanted to respond to. So I was saying that someone was asking that is a palmate leaf a simple or a compound leaf? A palmate leaf is a compound leaf because iko na several leaflets within it. So it should be a compound leaf, not a simple leaf. Then you take us to the next part, Mr. Paul. You scroll up so that we can be able to go through the one for the animals. Before I move to the one on the animals, there is arrangement of the leaves on the stem. On this part of the arrangement of the leaves on the stem, we have got the opposite. Alternate, that is when one leaf is not directly attached to the point, the other one has been attached. Such that I will be able to illustrate it within your screen. So if this is our stem, if our leaflet is within this side and another one within the same point, in oppositely arranged. But if it is this way, one is there, another one is on this side, another one this way, they are not within the same, they are not arising from the same point within the stem, then we refer to them as alternately arranged. And if we have got more than 2 leaves originating from the same point, and another one like that, there are 4 leaves originating from the same point of the stem, then we refer to that one as the whorled. So that is whatever we are supposed to get on that part of the arrangement of the leaves on the stem. Still, there is a question. Oh, that one I've arranged. What do you mean by bipinnate? Have mentioned or I've explained on the bipinnate. I've said that we have got the main leaf stalk, and within that main leaf stalk, it is branched. And within the branch, we have got the leaflets. I can repeat that. Tuko na this, hii ni main stalk, then there is a branch. Then the leaflets are arranged along this part. Many of those branches that way that have got leaflets within them. So that is what we refer to as the bipinnate. But for the pinnate, nimesema unakuwa na one main stalk na hapo ndio unakuwa na leaflets. Just that way. So that is the difference between those ones. Sorry. For the animals, these are the structures that we normally use in the construction of the dichotomous key. The type of skeleton that the animals have, we have got the endoskeleton, we have got the exoskeleton, then we have got another type of skeleton that you will be able to learn later in form 4 that involves the fluid. Like now within the worms, they have got the fluid type of skeleton within them. Although this one on a backbone, with or without, the backbone we do not prefer using this word. Instead, tunastahili kutumia the word notochord. Animal with the notochord, animal without the notochord. But in some of the questions, revision questions, whenever you go through them, you may get this one of the backbone. So it's one of the things or one of the areas that you're supposed to get. Of course, we know what it means from our primary school. What covers the body of an organism? Is it feathers? Is it fur? Is it scales? Is it hair? So we are supposed to use those ones in the construction of the dichotomous key. The locomotory structures. Which structures do those organisms use to locomote? Do they use the wings? Do they use the fins? Do they use the limbs in the locomotion? So we can also use them in classifying the organisms. Habitat that it occupies, we have got the aquatic, those ones that occupy the aquatic environment. We have got those ones that occupy the terrestrial environment. And also we have got those that are arboreal. Arboreal are still terrestrial. Arboreal are still terrestrial that will be occupying within the trees. Yes, Hakim has illustrated, has given us those ones who wanted to understand the difference between pinnate na bipinnate. So the first one there, I hope you are within the charts, those ones wenye wako bado na challenge. Unaona hapa ni only one stalk within the pinnate. The second one, iko the same way, but sasa it's branched. The main stalk has branches within it. So that is the difference between the pinnates na bipinnate. Thank you, Akim, for that image that will assist us to get better on that part of the pinnate and the bipinnate. Then we have got the text of the body. On the text of the body, we have got the smooth or the rough. Like now, if you will be able to hold the fish, its body surface is normally smooth. But if you if you hold, let's say if you hold a tortoise, it is normally rough. So you can be able to use the text of the body surface to differentiate. You can use the number of body parts. Some have got 2, some have got 3 body parts, especially for those organisms under the arthropoda. So you can be able to use the body parts in giving or in construction of the dichotomous key. Then we have got the mouth parts. What do they normally use for feeding? The teeth, the beak. So it's not teeth, beak. It's teeth separately, beak, the mandibles, the maxillae, the proboscis, among other mouth structures. So we can be able to use those structures in the construction of the dichotomous key. A point to note, Mr. Paul, you can kindly scroll up. Whenever you are constructing your dichotomous scheme, always start with those characteristics that will group the organisms into two broad groups. So usiende kuanza na the unique characteristic within an organism. Like now you are using maybe the mouth parts or the habitat that that animal occupies. And maybe within the group of organisms that you have given, that is the only unique characteristic for a single organism. So start with those ones that will group your organisms into two broad categories. Then continue until that will be able to group the organism or will be able to be specific for a given organism. Then the statements that we normally use in the construction of the dichotomous key, they are supposed to be contrasting. That is one statement the nini within, like now even unatumia the leaf type, within the first part unasema maybe leaf simple, then second part leaf compound. Of course huwa tunakuwa na pairs of statements. Because of those two pairs, that is why we refer to it as the dichotomous. Di means two. So two contrasting statements that should be short and simple. So we have given an example there. Leaf simple, you go to 2. Leaf compound, go to 3 or 4 that way. Have avoided to indicate 3 because it's just a portion of the dichotomous key that I've picked. I will be able to forward you one of the dichotomous keys that are selected. But if we'll have time, I will be able to, or we will be able to discuss that dichotomous key together and understand about it. So that is on the dichotomous key and the way or whatever we are supposed to consider on that. Take note of these also whenever we are constructing the dichotomous key and you are given specimens which have been given letters. We do not know the actual names of those specimens. I remember there is a year the students were given the jacaranda leaf, they were given the jacaranda, the rantamara leaf. In fact, it was a twig, the jacaranda twig, the Rantanakamara twig. Then we have got the other ones, which was which I cannot be able to remember clearly now. Then the students were supposed to use them to construct a dichotomous key. Then under the identity, remember those specimens were given letters that represents them. Students were indicating at the identity Rantana Kamara, thus indicating the jacaranda, and they lost marks because of indicating that. So use the letters given. Even if unaangalia unaona hii ni kel, usiandike kel, but indicate the letter that has been used within that part. There is someone who is asking, could you explain the difference between the mandibles and the maxillae? Normally, these mouth parts, even if I mentioned them, we do not go into the details of how they look like and their specific uses. But whatever we should get is that the mandibles are normally used to cut solid food substances. When insects are feeding on the solid food substances, they normally use the mandibles. Then the maxillae are fluid food materials that they will be able to obtain from the surface or from other animals. Kuna ingine yenye huwa tunaita the proboscis, like that one of the mosquito that it normally uses in sucking blood from organsyls. So hiyo ndio the part that we are supposed to get on that part. Let's move to the next part, which is the reproduction. For the reproduction, I want to move directly to the common areas that we normally get on these. We normally use the fruits for this process of reproduction. And you may be given a fruit and you are supposed to identify that fruit. We have got, like now, the type of fruits. We can have some fruits being berries or the Hesperidium. We have got the pods, we have got the pome, we have got the drupe. Like now the avocado, the mango, the one-seeded fruits, we normally refer to them as the drupes. There is a difference between the Hesperidium and a berry. And I want to hear from you the difference between these two words, Hesperidium and berry, although we normally mark them similar. When a student has indicated Hesperidium or the berry, the student can still score. But what is the difference between those two names, Hesperidium and berry? Someone to respond to that? A volunteer? A volunteer to respond on the Hesperidium and berry? Any try on that? Levi? Levi, can you try?
Sorry, but I don't know.
Yes, Levi.
I'm sorry, but I don't know the answer.
Oh, Levi anasema he doesn't know the answer to that. Someone else? Yes, Winnie?
A trial.
Yes.
Can you say Hesperidium?
Yeah.
A Hesperidium, it's not the endocarp, the mesocarp is white, fibrous, and fused to the epicarp.
Mhm.
While a berry, it has a succulent pericarp and it's simple and a fleshy fruit with many seeds.
There is a good trial. There is a good trial. Then you wanted to respond, Brian.
Okay, a berry is a fleshy fruit with a soft, fleshy pericarp, while a Hesperidium is a berry with a tough.
Brian, once more, repeat.
Okay, a berry has a soft, fleshy pericarp, while a Hesperidium has a tough mini leathery ring that contains aromatic oil.
That is a good trial. In fact, chenye nilikuwa nataka muemphasize, you have tried both. The epicarp. The epicarp for the berry is fleshy or succulent, but for the Hesperidium, it's hard or tough. I can give an example of a berry is a tomato. If you look at the epicarp is the outermost layer. It's normally fleshy. But for the Hesperidium, I can take the example of a lemon or an orange. It's normally tough. In fact, even you use this, that what we normally consume, we consume even the epicarp for the berries, but for the Hesperidiums, we do not consume them. They are normally tough. So fleshy epicarp for the berries, but the Hesperidium do not have the fleshy epicarp. It's normally hard or tough, the way Brian has explained it. The drupe nimeelezea, it's one-seeded fruits, like now the apples. No, like now the mangoes and also the avocados. They are the ones that we classify under the drupes. I've not indicated it within that list. The palm, hapo sasa ndio tunakuwa na apples within that part, I mean. The pod ndio tunakuwa na the peas, the beans, among other fruits there. The other part that I want us to look at is the type of presentation. Mr. Paul, kindly. Type of presentation. We have got different types of presentations. Ukiulizwa what is presentation, I really also want to hear from you. Let's share the meaning of this word presentation. Ukiulizwa, define that term presentation. What will you say? Nataka kusikia from you, the students. You try to respond to that. A volunteer. Yes, Winnie.
You can say a placentation is the arrangement of the ovules in an ovary.
Arrangement of the ovules in the ovary. Onchonga, I can see you also wanted to respond. Onchonga.
There's no difference with what she said.
Yes.
She said the same thing I wanted to say.
Oh, thank you. So that is exactly whatever we are supposed to give, that the arrangement of the ovules. So it's not the seeds. Although whenever we are making those sections, we normally observe the seeds, but the seeds have been formed from the ovary, or from the ovules, I mean, from the ovules. So that is why they can go to give us the way those ovules were arranged before the fruit was or had ripened. Mr. Paul, you can scroll for us up within that screen. So on this part of the type of presentation, we have got the various types of presentation. Meenda mbali kidogo, turudishe juu tena. We have got the various types of presentation, Mr. Paul. We have got the basal presentation. That is when let's go with the order. Turudishe juu kidogo, Mr. Paul. Tulikuwa tunaanzia on that presentation. That is point number 2 on reproduction. Yeah. Thank you there. So on the type of presentation, we have got the axial presentation. When when you make a transfer section, of course, we have got 2 types of sections that can be made when we are cutting our specimens. We have got the transfer sections, then we have got the longitudinal sections. In most cases, we make the transfer section, but we should not rule out that there is no time that we will be asked to make a longitudinal section. Though in most cases, ni transfers, but you can also be asked to make a longitudinal. For the longitudinal, especially for those ones that are in form of a drupe, unaweza ulizwa. You make a longitudinal section, then you draw a section of that. So when we have got, or when you cut, and that structure that we will get, that the seeds are arranged just like a wheel, that is what we refer to as the axial presentation. It has got the rocules, or the roculi, the partitions, and we have got the seeds within each portion. So whenever you are having it that way, that is what we refer to as the axial presentation. The marginal, like now that one of the the peas, the beans, they show the marginal presentation or the pods, the general name for those organelles there. The parietal, the placenta is located within the sides, within the walls of the fruits. Like now, the pumpkin, the passion fruit, they have got this parietal presentation. Then we have got the frecentrial. The placenta is on the central region, and the ovules are attached to this central placenta. We refer to that type of presentation as the free central presentation. Then we have got the basal presentation. On the basal presentation is when the placenta is located on the lower part, that is on the base. Then the ovules are arranged within the placenta that is on the base. Hakim anawaonyesha that. Yeah, that is good, Hakim. That is good. So you can see the marginal is on the first part. Then we have got the axile. So you can be able to see it's just like a wheel. It has got the roculi. Those partitions are the ones that we refer to as the roculi. So the tomato, the orange, the lemon are the ones that normally have that axile presentation. The axile presentation can also be referred to as the central presentation, but the common one that we normally use is that one. Parietal, you can see this, the ovules are arranged at the periphery. Hata wamekuelezea hapo at the periphery. An example is the passion fruit. We have got the pumpkin. Whenever you are cutting it, you can be able to observe that. We have got the free centriole. That is at the center. The placenta is there within the center. Then the seeds or the ovules are arranged on just across or around that placenta. The basal is there. I can give the basal is like that one of the mango again, or the avocado. Then this one, which we are having there, the superficial at all over the surface. So that is the one that we refer to as the superficial. I think we can give the guava, the mapera, are the ones that you can go to give us the best example. Akim, that is good. Thank you for that illustration. We can continue. Mr. Paul? Yes, Lucy. What is the presentation of a watermelon? Watermelon huwa tunaipea the axile presentation. If you look at the watermelon, it has got partitions that are very narrow, very tiny. So it's not something that is continuous. So we normally give it as the axile presentation. So that is what we should get there, Lucy. Mr. Paul, you can scroll up so that we can move to the next part. We have got this other part which is on the mode of dispersal or agent of dispersal. When you are giving the agent of dispersal, we only mention that agent. Like now, we can say animal, we can say wind, we can say self-dispersal. Yes, there is a question. I can just respond to that before we continue with that. Why can't we say marginal? Marginal for watermelon, not possible. Marginal is only for the pods. Pods are the ones that will contain marginal. So hatuwezi sema Winnie marginal presentation for the watermelon. Watermelon is a berry, and berries huwa zinakuwa na oxide presentation. In fact, I can recommend this, that now that you are at home, you can be able to obtain those fruits. So you can be able to go maybe tomorrow, take one sample each, then you can be able to cut transfer sections, and you be able to identify which type of presentation is that within the fruit that you have obtained. So it's something that we can be able to do at home. So let's try and purchase those fruits. Others, you do not even require to purchase them. They are at your homes. They normally grow within your homes. So can you try to get as many of them and be able to carry out that experiment, so that you will be able to understand better on that part of presentation. So I continue on this part of the mode of dispersal, or the agent of dispersal. I've already said if it's the agent, you only mention that agent directly. But ukiuliza mode of dispersal, you're supposed to say animal dispersal. Repeat that word dispersal again. Animal dispersal, wind dispersal, water dispersal, self-explosive mechanism, or self-dispersal. That way. So the word dispersal should be repeated whenever we are mentioning about the mode of dispersal. The reasons for the mode of dispersal, like now for the animals, we can say that the fruit is scented. Do not say, we cannot be able to taste the scent and say whether it's sweet or sour. So let's use just scented. Fruit, it is conspicuous, it is brightly colored, it is succulent, it's large. Those are some of the reasons that we normally give whenever we are giving this animal as the agent of dispersal. We can also give wind dispersal as another way that the seeds can be dispersed. We normally look at the wing-like structures within them. We normally look at their size. They are normally small and light. They are feathery, or they have got hairs within them. That is for wind. We have also the self-explosive mechanism, or the self-dispersal. On the self-dispersal, tunaangalia the lines of weakness, the searchers. Here, I want to emphasize this, that it's lines of weakness. It's not one line that is there. There are two lines of weakness that will enable it to burst and throw those seeds to further distance for them to grow or to germinate. So that is whatever we are supposed to get on these parts. When we are making the drawings, these are some of the things that we are supposed to consider. Our outline should be continuous. Had I made diagrams on these, but because of time, I will not be able to illustrate them. I will be able to post them later so that you can be able to see whatever is expected. So whenever we are making those drawings, like now on those diagrams that we have mentioned, all on those presentations that we have mentioned, we are supposed to ensure that we have a continuous outline. There should be no shading. Like now when you are indicating the juice sac and you show it all over, you lose a mark. Maybe you can show two inverted V's so that you can represent the juicy sac within a small section to show that it is all over that fruit. The same to the scales. Whenever we are using the scales, when we are drawing the fish, also we are supposed to show only three. tusiweke mingi. If it will be that way, then it will be shading. The shapes of the structures should be maintained. Like now, if you are drawing a seed and it was round, you should maintain that shape. Or if it's if it is oval, maintain that shape. The label lines to touch the structures are no arrows on them. So you should not show the arrow within those label lines. They should just touch that structure without hanging outside that. Then another thing is that whenever you do not, if you do not touch that structure, then you lose the mark there. The label lines that are pointing to a structure, they are not supposed to overlap one another. If they overlap, you lose both marks for the structures that you are supposed to label there. So avoid overlapping of those label lines within that part. Then the magnification should be indicated. Normally, whenever we are guiding our students, we normally guide them that they should maximize their marks on this part of the labels, because sometimes the student can mess on the drawing. That is that makes them to lose the drawing mark. But labeling of the structures, even the examiners are very flexible. So students, let's be keen. Ensure that you label the structures correctly. The label lines should not have arrows. They should not overlap. They should touch the structure. So, and they are the ones that normally carry a lot of marks within the drawings. Hiyo mark ya proportionality, mark ya correct drawing, those ones, maybe you can try your level best to get them. But if you do not get them, ensure that you get marks on the label lines. Proportionality, not all the times that they give that mark, they normally tie or combine that mark for the proportionality together with that one of the drawing. That is what we can give on that. I want us to move to the next part, which is on the cell physiology. Mr. Paul, kindly. You scroll for us our screen up so that we can go to this part of the cell physiology. So on the cell physiology, these are the main processes that are normally tested. We have got the osmosis, then we have got the diffusion. These are the main ones that we normally get during the process, the cell physiology. The main substances that are normally involved, they are the plant tissues, the Fisking tubing, the solutions, like now you can have the distilled water, you can have the sugar solutions. There is Mr. Paul, there is a photograph that I had sent to you. If you can kindly, a photograph that was having this experiment on the cell physiology, so that we can be able to discuss with the students. So while we are waiting for that, I can give this information that the questions on the cell physiology, we are supposed to be very keen. Those terms that we normally use there, they make us to lose marks. Like now, we cannot say a plant tissue is turgid or flaccid. What becomes turgid or flaccid is the cells within the plant. So whenever we place a plant tissue within distilled water, in this case, distilled water will be an hypotonic solution. The cells of the plant are going to gain water and become turgid. So it's not the tissue that is becoming turgid, but the cells of that tissue, of the plant tissue, are the ones that becomes turgid. Then, if we place it within an hypertonic solution, like now that solution is having high concentration of salts. So the cells of the plant will lose water and become flaccid. So it's not the tissue that becomes flaccid, it's the cells. But how do we describe those tissues? If the tissues of the plant gain water, if the tissues of the plant gain water, they will enlarge. They will enlarge. They will lead to the increase in size of the tissue. Yeah, thank you. There it is. So they will enlarge, and the size of the plant will increase. We can also observe the curvature when we are dealing with that part. Within your screens, you have got a section of fruits that was immersed into solutions. The first solution is distilled water. The second solution is 50% salt solution. Then those structures that were placed within those solutions were the banana peels that are 2 centimeters long. So they were placed there, then they were left for 30 minutes. Then the observations that are supposed to be made are as shown there. I wanted to get within this part. A banana peel is a fruit. And when we are cutting the banana, we have got the green part, which forms the apical. Then we have got the white part, which forms the mesocarp. So, yes, that way, Mr. Paul, it's okay that way. I can be able to explain it that way. So, the white part, is the white part is the mesocarp, while the white part is the mesocarp, while the green part is the epicarp. Normally, this epicarp is waterproof, so it's not affected by the water. Whenever we immerse this peel within the water or within the distilled water or whichever solution, those cells within that part, within the epicarp, they are not normally affected. But the cells of the mesocarp, they can allow the movement of water in and out. So that is why, if you can be able to look at the first section there, the observation that is made is that these cells of the mesocarp have enlarged. And because of them enlarging, that is why it's curving inwards, the way you can be able to, the way you can be able to observe there. So that is something that you're supposed to note within that part. Then the other one is that if you place the same section within the 50% salt solution, then it means that those cells that can allow water to pass or to get into them are the ones of the mesocarp. So they will lose water to the salt solution because the salt solution is hypertonic. Then they will shrink. And that makes the mesocarp, to curve inwards. So for distilled water, mesocarp, curves outwards, while in the hypertonic solution, the mesocarp curves inwards. So that is something that we are supposed to get. So it's not all the times that the students should use the word the epicarp and the cortex or the cortical cells. Of course, for the stem, Those are the terms that we are supposed to use, the cortical cells and also the epidermal cells. So for the fruits, the words that we use is the epicarp, the mesocarp. Epicarp, the cells within the epicarp, not affected by water because they are waterproof. Cells of the mesocarp, they allow movement in and out of water molecules. So they can be affected by water. That is why it is the mesocarp that will be responsible for the two curvatures that we are able to observe within our diagram there. Mr. Paul, take us to the main document. So that is on that part. So in case you are given for the stem, use the word cortical cells and the cortical cells, and the epidermal cells. If you are using the fruit, use the epicarp and the mesocarp. Have given the way you expect the mesocarp and the epicarp to be affected by the water. I can take the questions. It's okay. Yeah, Mr. Paul, move the document so that we can be at the microscopy. That is the last part that I want us to discuss. Yeah, that is the part. Thank you. So we did this part on the microscopy. Like this year, the students were tested on this part of the microscopy. What are we supposed to get, or what are we supposed to observe whenever we are doing the microscopy? Students should be conversant on how to use the microscope or to handle the microscope. When we are cutting the specimens that we are supposed to place within the microscope, we are supposed to know the correct way that we are supposed to do that. Like now, in this year's examinations, the students were supposed to peel a part of the epidermis of the cells and place it within the microscope. Then observe the stomata and the epidermal cells. Most students had a challenge on that, on how to peel the epidermis of the cells. So you should understand on how we should remove the membrane on the lower part or the upper part of the plant, and you mount it within the slide, or you place it within the slide and place it within the microscope so that you can go to observe the structures required. Then now, after you have placed your structures within the microscope, the next thing that you're supposed to do is that you're supposed to make your observations. So I can advise you, my students, that you are supposed to make the drawings, make observations and make drawings from the structures that you will be able to observe. My advice is this, that get the main structures that you have been asked to draw. The other parts, the other structures that you may be seeing, you may not be able to draw them because the examiner do not require them. So the moment you place those structures there, it will be treated as shading. And uonyeshe kama ni the stomata or the guard cells, you show how they look like when you have used your microscope. Then after that, you will be able to label them. I said, anywhere where you are making the drawings, maximize your marks on the label lines. The same, there is a year that the students were required to cut a transfer section of the peshore of a reef, then observe it by use of the hand lens and also by use of the microscope. So also for that, you are supposed to look at those main structures you capitalize on that. You draw them, then you label them. So avoid drawing those other things that you might be seeing. Unaona tu dots twingine, maybe because of the stain, then you want to illustrate them within your diagram. That students and further we are very keen and we only emphasize on that part that is required and you label it correctly. Label lines have said touch the structure, not harm, and they should not have the arrows, no overlapping. We should also get on how we get the magnification. Of course, we know that the magnification is the multiplication of the eyepiece lens magnification. Within my statement there sijaandika eyepiece lens magnification. Of course, tunajua ni lens, si eyepiece, tunaangalia magnification yake. Eyepiece lens magnification times the objective lens magnification. Students huwa wanasahau kuandika magnification, always ensure that you indicate it. Eyepiece lens magnification times the objective lens magnification. In the microscopes that we use in our schools, the eyepiece lens huwa inakuwa ni times 10. Objective lens depends on the type of objective lens that you use. Sorry. Of course, tunakuwa na the 3 categories. Low power, yenye inakuwa ni times 4. Medium power times 10. The high power ni times 40. So, tunatumia hizo. The linear magnification, if you are using hand lens, you use this formula for the calculation of the magnification. You take the length of the drawing, you divide by the actual length. So you have observed the organsim and you have drawn. You can measure the length of that drawing, then you divide it by the actual length of that organsim or specimen that you are using. So that is how you use the linear magnification. So mainly, ukitumia hand lens uambiwe show the magnification or how you obtain the magnification for that. That is whatever we are supposed to do. Other areas that we can get that we have not discussed is about the enzymes. Here, on the enzymes, although we'll be able to discuss them later in a different session, the enzymes, we normally look at the conditions under which these enzymes will be working best. Like now, in terms of the temperatures, very low temperatures, very high, and the optimum temperatures, we look at the pH of those of the solutions in which those enzymes will be working on. So that is what we check on that. So I've not gone into the details of that. I've not gone into the details of the practicals that involve the evolution. Evolution mainly is in Form 4, and we have got the practical activities on the evolution that you can be able to get in an exam. We have got the genetics, also a topic in Form 4 that can also be tested in the practicals. We have got the ecology. So because of the limited time, I will not be able to exhaust on those others. So I've only given a summary, but the details I will be able to discuss them in another session. I will be able to give the details and also the examples. So thank you for your time. If there is any question, you can be able to ask through the chats, and I wish you well. Have a good night. Mr. Paul, if you can get me back to you.
Thank you, Bwana Makori. Thank you for the good lesson. It was an interesting lesson, and I thank God that at least we have gotten much. I have seen Mr. Mutiso in the class. Mr. Mutiso, if you are there, you can say hello. Mr. Motiso, kindly. Mr. Motiso, if you can hear me. All right, I think Mr. Motiso has gone. So, Mr. Motiso. All right, thank you. So thank you, students, for that lesson. It has been good, and thank you because we have paid attention. Now, I want to remind you that we are having actually the English lessons, and you have seen how we're supposed to subscribe. Kindly make sure you do that. We already read chapter 1 of Blossoms of the Savannah, and those that managed to log into the class so that it was an interesting lesson. So I want